Hi there guys, and welcome to another A-level biology video. Mr. Phillips. This is for year 13, the respiration or the energetics module, I suppose, respiration and photosynthesis, and I believe it's video 5.5. And just to remind you, I'm teaching respiration before photosynthesis because I think that helps you understand photosynthesis much more. So let's go on to it. Anaerobic respiration. So just some key words which we've dealt with over the last few lessons I thought would be worth reminding you of. So if something is phosphorylated, it means we add a phosphate group to it. If something is dephosphorylated, it means we remove a phosphate group from it. If something is hydrogenized, we add a hydrogen. If something is reduced, a hydrogen or electron is added. If something is oxidized, a hydrogen or electron is removed. If you remember the saying oil rig, oxidation is loss, reduction is gain. Aerobic means you're using oxygen. Anaerobic means without oxygen. So it doesn't mean with very little oxygen, it means without oxygen. And then decarboxylation is the removal of a carbon, or it is the removal of a carboxyl group. Now obviously when you remove a carbon it makes carbon dioxide, but you don't say the removal of carbon dioxide. Okay, so what does anaerobic respiration mean? So some questions you should be familiar with. So anaerobic respiration means there is respiration in the absence of oxygen. So again, it doesn't mean in little oxygen, it means without oxygen. What does anaerobic respiration produce in animals? You should know from GCSE that it produces lactic acid, which we might also refer to as lactate. What does anaerobic respiration produce in plants and yeast? You should also know now from GCSE, and that is it produces carbon dioxide and ethanol. And then which parts of respiration are affected by there being no oxygen? Well, it affects really the last stage, the electron transport chain, oxidative phosphorylation. No oxygen, the electrons can't be accepted. That then shuts down the Krebs cycle and the link reaction. And the only stage that can occur in the absence of oxygen is anaerobic respiration. It's glycogen. It is the glycolysis. Sorry. So let's have a look then, just to remind you about the final stage of respiration and how it worked. So if we have a look here, we have the oxygen. So the whole point of oxygen is it is the final hydrogen and electron acceptor. So if we get rid of that oxygen, what won't happen is these electrons in this cytochrome won't be able to be passed on to the oxygen. That means if they're trapped in there, these electrons won't be able to be passed along, okay, and the one electrons in cytochrome 3 will stay. The electrons then in this cytochrome will stop in there, and so on and so on. So if you don't pass the electrons from one cytochrome to the other, if that passing stops, what that means is the energy isn't going to be released, and that means that these hydrogens will not be moved across the membrane, and you will stop producing ATP in this stage. It also means eventually with the electrons that the electrons won't be able to be taken away from the NAD, and it will stop the NAD becoming oxidized. So we need, for anaerobic respiration to happen, all of this stage can't happen, so you're losing all these ATPs that have been generated, but also for anaerobic respiration to happen, we have to find a way of oxidizing this NAD. Otherwise, you'll run out of NAD, and therefore you won't be able to continue the first stage of respiration, glycolysis, where you need NAD to take away the hydrogens to break glucose into pyruvate. So what happens if the oxygen runs out then? So as I just sort of summarised then, oxygen is the final electron acceptor in oxidative phosphorylation. So if it disappears, if there's no oxygen, electron transport chain stops. That means there'll be no new NADs or FADs, but when we focus on NAD, that are oxidized. So therefore the Krebs cycle and the link reaction will also stop. Only glycolysis can happen, but in order for that to happen, we need another way of oxidizing those NADs so we can reuse them and allow them to reduce the glucose into pyruvate. Now we should know from GCSE there's a lot less energy in anaerobic respiration, so you only get 2 molecules of ATP yield from every glucose. So if you remember the reaction from glycolysis from the second video I did on this subject, that produced 4 ATP but only a yield of 2. And the glycolysis can happen because of two types of reactions. So plants and fungus use ethanol fermentation, which you should know from GCSE, and animals use lactate fermentation, which you can also call lactic acid fermentation. So let's have a look at it. So these are two flow diagrams that sort of show us what happens, and they answer these questions for us. So first of all, you should know it's animals that make lactic acid or lactate, but particularly mammals. So you might get asked in an exam mammalian respiration or animal, but effectively you can assume it's the same thing. So just to remind you what has happened, we've had glycolysis occurring. So this diagram here, there's two diagrams that are sort of separate here. So there's one on the left and right of that line. Okay, but this reaction here, this bit shows you glycolysis. Okay, and in order for glycolysis to keep occurring, we have to remove hydrogens, which causes reduced NAD. That reduced NAD will only last a certain time until it is all reduced, however, so it has to be reoxidized. So what we're saying is we have to take the hydrogens that are in those NAD and we have to put them somewhere else. So we take the hydrogens out of the NAD and we insert them into something here to turn pyruvate into lactic acid or lactate. That then reoxidizes the NAD, and the NAD can then go back into the reaction to collect more hydrogen and to allow the glycolysis reaction to keep happening. But what is accepting? How is it accepting those hydrogens? So if we look at this graph, equation effectively, which answers question 2 of your on the slide there, what is happening in this reaction? Well, what we're doing is we're starting off with the molecule pyruvate. Where does that come from? Well, just remind you, it comes from the end of glycolysis. We need to do something with that, and we use it to get those hydrogens off the NAD. So the reduced NAD, which is also made in glycolysis, and we need to reoxidize so we can recycle it and take it back to glycolysis. So the NAD, as I just said, sorry, just to pause the video for a second, goes back into glycolysis to pick up more hydrogens, and then that cycle can continue. But in order for that to happen, we have to take the hydrogens out of the NAD. We have to oxidize the reduced NAD again. So how does that work? Well, what we use is an enzyme called lactate dehydrogenase. So the lactate dehydrogenase basically takes the hydrogens off the NAD. So the enzyme I'm talking about here is this one, lactate dehydrogenase, and it takes the hydrogens off the NAD and attaches them to the pyruvate, okay? And that converts then pyruvate into lactate. And that's that. Now what that leaves us with, though, unfortunately, is a lot of lactate, and as you should be aware, lactate or lactic acid is an acid, so it causes muscle fatigue because the enzymes start to get affected by the wrong pH, start to denature, and that then leads us to an oxygen debt. And when we breathe in oxygen afterwards, when you finish exercise, you have to breathe in that extra oxygen in order to break down that lactate. But let's quickly go on to alcohol fermentation. So alcohol fermentation or ethanol fermentation happens in plants and yeast, and this is effectively how we make alcoholic drinks and how we make alcohol that we can use in fuel in different countries and different things like that. So what we've got is glucose. Again, this is just typical glycolysis happening as it happens in all organisms. We need to again get this reduced NAD and oxidize it. So instead of doing it making lactate, we do a two-stage reaction which turns pyruvate into ethanol and carbon dioxide. So let's have a look at the what's happening in this reaction and try and make sure we can understand it. So again, ignore all the chemical symbols for pyruvate and ethanol. You don't need to know those. But effectively, if you take So this pyruvate, what we need to do with the pyruvate is the pyruvate has an enzyme act on it called pyruvate decarboxylase, which hopefully makes sense, the name of that. That takes off from the pyruvate a carbon dioxide, which is emitted, so hence this produces carbon dioxide. That causes the pyruvate, once you've taken that carbon dioxide off, to turn into ethanal. Okay, so remember it's an A first for the alphabet. Okay, and then what happens is the ethanal is reduced, it gains hydrogens to become ethanol. Okay, and where did those hydrogens come from? Well, as you probably guessed, this is our method of getting the hydrogens off the NAD. So the NAD are oxidized, the NAD loses some hydrogens, okay, to make oxidized NAD. They can then go back to glycolysis, okay, to allow that reaction to continue. And the enzyme that does that is called ethanol dehydrogenase. And you might think, well, that's a bit strange. Why is it called ethanol dehydrogenase? Well, it is because it can't be called ethanal, because there's another enzyme that does that. Okay, why was this so useful? Well, it obviously continues to oxidize the NADs, which means that glycolysis can continue as a result. Okay, can it allow for the continuation of the electron transport chain? No, it can't, because there's still no oxygen, so then the electrons cannot be passed along the chain. Okay, and can organisms do this forever? Well, we'll talk about that in the next few slides. So back to lactate in animals then, so a bit more about that. So we've talked about the equation before. We've talked about oxygen debt, okay, and basically depending on the situation, when you breathe in that extra oxygen after you finish exercising, it's to remove the lactate, okay, and it can cause one of two things to happen. Some of the lactate can be converted back into pyruvate, okay, but a lot of the time, if you think about the original reaction, you would have been respiring with oxygen, you would have made two products. So the products you will make because you are adding oxygen to lactate is the original aerobic respiration product, sorry, water and carbon dioxide, which you can't draw, and that basically is what happens as a result of that. In plants and yeast, we obviously produce ethanol alcohol. Now, ethanol or alcohol is not a desired property for yeast or plants. It's a byproduct, a bit like carbon dioxide for us. So what happens with this is that yeast is what we call a facultative anaerobe, which means just like we are, it can survive for a while on anaerobic respiration, but it doesn't want to. And the reason it doesn't want to is because if the ethanol gets up to 15%, then it starts to kill the yeast cells. It will grow much faster than the yeast or the plants will under aerobic conditions, but it will survive under anaerobic conditions. And that's why alcohol, if you buy alcohol, the alcoholic drinks, before they've been distilled, the highest level you can normally get to is maybe 13-14% for like a wine or something like that. And that is because any higher, and it would actually start to kill the yeast that has produced it. Okay, so breaking down the toxins, we said both of them are toxins, okay, so they have to be excreted. Ethanol is just excreted, it's just released, and that's why the plants do it. And we said lactate can be turned back into in the liver, and what it will turn a lot of it into carbon dioxide and water again, or it will convert it back to pyruvate, or it can even convert it to glycogen. And obviously oxygen is needed for that, and that is called the oxygen debt. Now, just finishing off and making some comparisons, okay, so in terms of yields of ATP, if you remember, we said there is about for aerobic, which this first column is, there is about a 30 yield of ATP overall, okay. The oxidizer of the NAD, so what causes the NAD to be oxidized, is the cytochromes in the final stage. So I'm just going to write cyto there because I can't fit it in, and you need to know the name of the first cytochrome. Does glycolysis occur? Well, we should know yes, it does. Does the Lincoln reaction and Krebs cycle occur? Yes, it does. And obviously, does electron transport chain occur? Yes, it does. Is oxygen used? Yes, it is. And is a mitochondria needed? Well, yes, because the link reaction, Krebs cycle, electron transport chain all happen in the mitochondria. So we look at animals then. The lactate pathway, you get a yield of only 2 ATP per glucose, okay, and the oxidizer of NAD is the pyruvate, which then turns into your lactate as a result of taking the hydrogens away from the NAD. So I'm going to write pyru for pyruvate there. Does glycolysis occur? Yes, that's the point of it. Does this link reaction Krebs cycle occur? No. Does the electron transport chain occur? No. Is oxygen used? Well, it's anaerobic, so I doubt it. And is mitochondria needed? Well, no, because anaerobic respiration is glycolysis only, so that happens only in the cytoplasm of the cell. And the last one, alcohol fermentation or ethanol fermentation in plants and yeast, 2, okay, because the only 2 yield of glycolysis again. The final hydrogen acceptor or the oxidizer of the NAD, which takes the hydrogens off the NAD, is, I'm really struggling to write this, so I apologise, is your ethanol. Okay, so it's your ethanol. That's terrible, well do. Okay, does glycolysis occur? Yes. Okay, obviously we've said that. Does the link reaction Krebs cycle occur? No. Does the electron transport chain occur? No. Is oxygen used? No, because it's anaerobic. And is mitochondria needed? No, because it's anaerobic. So you get a lot less energy for anaerobic respiration. You have to deal with your problems of lactate or ethanol, which are poisonous, and the pyruvate converted to lactate is you're allowing the NADs to be oxidized so you can continue glycolysis, and ethanol being converted to ethanol takes that hydrogen away, which allows the NAD to continue. So I hope that's been useful, and I'll see you next time.